Good morning, dear students. In this class, we'll be seeing about bioremediation of heavy metals. So the different aspects of this topic are how microbes are used to transform the heavy metals and how certain microbes are able to tolerate the toxicity of heavy metals and what are the different types of metal microbe interactions. Then its application, how metals are immobilized and also transformed, and further applications like biomining and bioleaching of heavy metals using microbes. So when you talk of microbes, when you talk of these heavy metals have certain characteristics. And the very important characteristic why it is called as a heavy metal is that because it has a specific gravity which is more than 5 gram per ml. And the other characteristics are they are capable of cationic hydroxide formation, formation of complexes, and also certain heavy metals have environmental toxicity. Roane and Pepper have classified heavy metals or grouped heavy metals into essential metals. Essential metals are those which have known biological functions. For example, cobalt, manganese, iron, nickel, zinc, copper, molybdenum all have their role in biological cells. For example, iron, they are part of hemoglobin, and several other metals are also part of enzymes, coenzymes, etc. And next is toxic metals like cadmium, mercury, lead, which cause toxicity to the biological systems. And some of the heavy metals are non-essential, non-toxic metals with no biological effects, like cesium, strontium, etc. And metals commonly associated with pollution like arsenic, cadmium, copper, chromium, mercury, lead, and zinc. Normally, when we talk of heavy metal pollution studies, we talk of these metals or study the effects of these metals. And there's a need for metal remediation or removal of metal because these heavy metals are linked to certain toxic effects, like, for example, birth defects, cancer, skin lesions, retardation leading to disabilities, liver and kidney damage. And now, because of anthropogenic activities, we find that the concentration of heavy metals in places like industrial areas in our country are much greater than the or much higher than the permissible limit of WHO or any other standards. Because even for heavy metals, there are permissible maximum limits above which they cause toxicity to the ecosystem. The sources of heavy metals are mining, ore refinement, industrial manufacture of metal alloys, batteries, electrical components, paints, preservatives, insecticides, etc. Even paints have a lot of heavy metals. Heavy metals provide a luster or a shining effect to the paints. And there was an article in the Down to Earth, the magazine Down to Earth, regarding the concentration, a high concentration of heavy metals in several commercial paints. And the heavy metals have been used for a long period of time. And archaeological evidences in Israel indicates that the mining and smelting of ores for extraction of metals, including heavy metals, have been carried out since 4500 BC. The toxicity of metals depends mainly on the bioavailability. Certain metals are easily available even though they are present in the environment. Some metals may not be available, that is, they will be in the non bioavailable form. And some metals may be easily bioavailable. When they are bioavailable, the toxicity also increases. And the bioavailable forms are mostly The soluble forms, that is, they will be in the dissolved form, in aqueous form, and they will not be sorbed, non-sorbed form. They will not be sorbed to clay or any solid matrix, and because they are soluble, they will be mobile also. So in these conditions, the bioavailability increases. When they are soluble, when they are not sorbed, sorbed means they are absorbed to a surface, or they will be mobile. And the non-bioavailable forms are mostly they will be in the precipitated form, or they will be in the complexed form and absorbed form, and they will be non-mobile. No, they will not be mobile. The bioavailability depends upon the different pathways of the metals in the environment. And here you can see that the aqueous phase of a metal, when they are in the soluble form, they may be present in the surface water bodies, or through the soil they can go to the groundwater because they are more mobile, they are not soft, and they are soluble. So when they are present in the surface water, they are also capable of volatilization or moving to the gaseous form into the atmosphere, for example, mercury. And sometimes they can be precipitated as metal salts. When they are precipitated, they are in the non-bioavailable form. And these aqueous phase metals also can be formed as complexes, or they can be sorbed to minerals and also organic matter, and thus they can become non-bioavailable. And sometimes the aqueous phase metals can also be taken up by the plants, and they can move through surface runoff. And if they are leached, they go to the groundwater and they'll be present in the groundwater. So these are the different pathways of heavy metals in the natural environment. And the four essential factors that influence bioavailability are metal chemistry, cation exchange capacity or the CEC, the redox potential, and pH. Metal chemistry, when we talk of the metal chemistry, heavy metals can be, the chemical forms can vary. Cationic metals, for example, like lead, they strongly interact with the negative charges of the clay minerals because they have a positive charge. They interact with the negative charges of the clay minerals and anionic salts like phosphates and sulfates. And these cationic heavy metals are also attracted to negatively charged cell surfaces where they can be taken up by the cells and they cause toxicity. And the another factor is cation exchange capacity. And this cation exchange capacity or CEC depends on the soil organic matter and clay content of the soil. So the low CEC means low metal binding capacity and high metal toxicity. That is, they are exchanged for cations. And the humic substances which are present in the soil contain several functional groups like carboxyl group, carbonyl group, phenyl group, hydroxyl, amino, imidazole groups, sulfhydryl groups, and sulfonic groups. They interact with the metals and make them non-bioavailable. So they are sorbed to these, or they are form complexes which these functional groups, and they become non-bioavailable. Next important factor, the third factor is the redox potential. Redox potential means oxidation reduction potential. So each depends on the availability of oxygen. Under aerobic conditions in the soil or in the sediment, the redox potential ranges from 0 millivolt to plus 800. But when the oxygen concentration reduces, the redox potential also reduces to negative, that is from 0 to minus 400. that is highly reduced condition or highly anaerobic conditions. So in the oxidizing or aerobic conditions, from plus 800 to 0 millivolt, the metals are found as soluble cationic forms, like for example, Cd2 plus and lead 2 plus. And the reduced or anaerobic conditions or highly reduced redox potential that results in precipitation of the metals. For example, copper sulfide, lead sulfide. For example, in the anaerobic conditions, normally the sulfate reducing bacteria, it prevails under strict anaerobic conditions. So they reduce the sulfate to sulfide, hydrogen sulfide. And these sulfides react with the metals to form the different metal sulfides. And the fourth chemical factor is pH. So at high pH, metals are found. At high pH means at alkaline conditions, metals are found as insoluble metal, mineral, phosphates, and carbonates. And at low pH, metals are found as free ionic species or as soluble organometals. And as the pH increases, the metal solubility decreases. That means in the acidic pH, the metals are highly soluble. And the toxicity of metals. Why metals are toxic or how they are toxic? So metals are toxic because of their strong ionic nature. Because of their strong ionic nature, they bind to many cellular ligands and displace the native essential metals for the normal binding sites. For example, arsenic can replace phosphate in biological cells. Biological cells contains phosphate, and arsenic, if it is taken up by the cells, that will interact with the cellular ligands and it will replace phosphate. So wherever phosphate is in our cells, it will be replaced by arsenic. So the biological functions will be disrupted. Metals also disrupt proteins by binding to the sulfhydryl groups of the proteins. And they also disrupt the nucleic acid by binding to the phosphate or the hydroxyl groups of the nucleic acids. For example, cadmium competes with cellular zinc and non-specifically binds to the DNA, inducing single strand breaks of the DNA. You can see how the heavy metals can enter into the cell. This is the cell membrane, and this is outside the cell, and this is inside the cell. So you can see through lipid permeation, M power 2 plus shows the metal, heavy metal, anionic metal. The lipid permeation metal can enter into the cell. By endocytosis, the metals can enter into the cell. Using ion channels, also the metals can enter into the cell. Using ion pumps, carrier mediated or proteins are normally used as the carrier. Through this carrier mediator also, metals can enter into the cell, and also through complex permeated, metals can enter into the cell. And metals also causes some toxicity to the microbes. Even though some of the microbes can escape the toxicity, generally metals are toxic to microbes. You can see the different toxic effect. This is the bacterial cell and the different toxic effect of the metals to microbes. For example, metals like mercury, lead, zinc, nickel, copper, cadmium can disrupt the cell membrane. And you know the central dogma of biology, that is DNA to RNA to protein synthesis. And certain, already we saw about DNA, disruption of DNA by certain heavy metals like mercury, lead, cadmium, arsenic can disrupt the DNA and cause DNA damage. And mercury can inhibit the transcription process from DNA to mRNA, and the translation process of mRNA to protein synthesis is inhibited by metals like mercury, lead, and cadmium. Then enzyme activity is also inhibited by heavy metals like mercury, lead, arsenic, cadmium, and copper. And protein denaturation occurs by heavy metals like mercury, lead, cadmium. Cell division is also inhibited by certain heavy metals like lead, cadmium, mercury, and nickel. And certain microbes have evolved strategies to overcome the metal toxicity and cause some resistance. So the strategies or the methods of resistance are of two types. Either they prevent the entry of the metal into the cell, they can, in the cell membrane itself, they prevent the entry of the metal into the cell, or if it enters into the cell, they can actively pump the metal out of the cell. And for this, they have several strategies, like for example, sequestration. Sequestration means capturing. This occurs by metal complexation with extracellular polymeric substances and also metallothionein like proteins. So, the microbe will be capable of capturing the heavy metals. That is called sequestration by means of complexing the metal with certain substances which are produced by the microbe, like extracellular polymeric substances and metallothionein like proteins. They are also called siderophores. We'll be seeing about this later on. What are these in detail? This first mechanism is sequestration or metal complexation. Second is active transport, that is using ATP pumps, APT dependent efflux pumps. First sequestration will be to prevent the entry of the microbes into the cell. And if it enters into the cell, it will be pumped out by means of ATP dependent efflux pumps. And the third is oxidation reduction. Microbes will either oxidize the metal to make it more mobile and move it out of the cell, or it will reduce the metal to immobilize the metal and make it non bioavailable so that the toxicity will be reduced. So these are the different strategies of a microbe to resist the metal, to either to prevent the entry of the metal into the cell, or if it enters, to um, prevent the or to reduce the toxic effects. So you can see given in a nutshell, this figure shows in a nutshell what I have told earlier. So this is the blue color is the cell membrane. This is the black is the flagella, and the yellow is the extracellular polymeric substances that is produced by certain microbes. Not all microbes. Certain microbes can produce certain polysaccharides. Actually, this is produced to prevent the microbe from dying against desiccation in adverse conditions of water unavailability. The extracellular polymeric substances or the sugary substances which are produced by the microbes prevents the cell against desiccation. And this EPS or extracellular polysaccharides sequesters or captures certain heavy metals outside the cells and thus prevents the entry of this heavy metal into the cells. That is EPS sequestration. That is, for example, cadmium. And certain microbes will also bind the heavy metals on the surface of the cell membrane. Okay, that is called cell wall binding. And certain microbes are able to precipitate the metals as salt. Already I told you, cadmium, sulfide as sulfide salts or phosphate salts. They form salts with the non-metals. And certain heavy metals are reduced. For example, mercury is reduced, copper can be by changing their form to a reduced form. Then metallothionein like proteins which are produced also forms complex with the metals, and certain ATP driven pumps can pump the metals which enter into the cells. They are pumped out of the cells, and certain metals can be volatilized. For example, mercury can be volatilized. All these are certain strategies of how a microbe can prevent the toxicity of heavy metals. And some of these strategies are used to immobilize the heavy metals and also remediate, bioremediate or remove the heavy metal from, for example, from the environment, also from the wastewater. So microbial interaction with metals, oxidation reduction reactions, chromium 6 to chromium 3, for example, Pseudomonas fluorescens, reduce the chromium. Chromium 6 is highly toxic and chromium 3 is less toxic. And this microorganism, bacteria, Pseudomonas fluorescens, is able to reduce the chromium from chromium 6 to chromium 3. And complexation, non-specific binding to the cell surfaces, or they are taken up intracellularly. Then methylation results in volatilization and increased toxicity of the metal. We have heard of the Minamata incident where the mercury was converted to methyl mercury. That becomes more toxic. That is, it is capable of bioconcentration and capable of transferring from one trophic level of the food chain to the other. That is called biomagnification. So sometimes it increases the toxicity also. Then we have other substances like biosurfactants, bioemulsifiers, and siderophores, which helps. These are the biosurfactants, bioemulsifiers, siderophores, or the metal chelating proteins, are those which are produced by the microbes extracellularly and they interact with the heavy metals. So, what are extracellular polymeric substances? EPS, exopolymers or extracellular polymeric substances, are composed of carbohydrates, polysaccharides, and sometimes nucleic acid and fatty acids. EPS are efficient in binding the heavy metals like lead, cadmium, and uranium, and the EPS binding is pH dependent. And this is used to immobilize certain heavy metals, for example, immobilization of lead by Staphylococcus aureus, Micrococcus luteus, and Acetobacter species. Next is the siderophores. Siderophores are iron complexing, low molecular weight organic compounds. And these siderophores interact with other metals similar to iron, such as aluminum, gallium, and chromium. That is, these siderophores are produced by microbes to chelate the iron from the surrounding environment and use it for the microbes during adversity times, that is during the unavailability of iron. This iron, which is complex by the siderophores, will be used by the microbe. And this capacity to complex iron is also used to complex other metals like chromium. Siderophore complexation reduces the copper toxicity in cyanobacteria. Next is biosurfactants. Biosurfactants are like, for example, soaps or emulsifiers. So increase the uptake by microbes can produce biosurfactants, increase the surface area, and increase the availability or uptake of heavy metals. Biosurfactants can increase the solubility of metals by increasing the surface area, and it can complex the metals like cadmium, lead, and zinc. So these are the different mechanisms of interactions of heavy metals.